let's look at the functions of carbohydrates. The functions of carbohydrates. What are the functions of carbohydrates? So you should be able to know it. Most abundant dietary source of energy for all organisms. Participate in the structure of cell membrane and cellular function. Structural component of many organisms, including the fiber, cellulose of plant, exoskeleton of some insect, and then does the work of storage of energy. So you see, carbohydrate has how many functions? 1, source of energy. 2, structure of cell membrane. 3, involved in cellular recognition, cell to cell recognition. Alright. 4, they are involved in formation of they are involved in storage function, so that the cells can meet immediate energy demands of the body. So what is the classification of carbohydrates? Like we already know, carbohydrates are classified into monosaccharides and oligosaccharides, and then polysaccharide. So we have monosaccharide, which are example glucose, fructose. Oligosaccharides are disaccharide, trisaccharide, tetrasaccharide, pentasaccharide, up to 9 or 10 decasaccharide. Alright, but above that you have polysaccharide. Now there are 2 types of polysaccharide generally, but another name for polysaccharide is glycans. Alright, there are generally 2 types of polysaccharides: the homo polysaccharides and the hetero polysaccharides. What is homo polysaccharide? Homo polysaccharide contain only 1 type of monomer. On hydrolysis, they yield only 1 type of monosaccharide. Example: starch, cellulose, and glycogen. What makes them monosaccharide? Starch contains many glucose joined together. Many glucose joined there together. So because it's only glucose that is in it, it is called homo. The same sugar. That's why it's called homo polysaccharide. Also, the same thing cellulose is only glucose. The same thing glycogen is only glucose that is there. Alright? But something like hetero polysaccharide, these ones, they contain 2 or more types of monomer. On hydrolysis, they will use mixture of monosaccharide. Example is hyaluronic acid, chondroitin sulfate, heparin, and mucins. So all these ones, they are, you can see homo polysaccharides, all of them is yellow. All the monomers are yellow. But if you see heteropolysaccharide, you see that some are pink, some are blue, okay, some are green. So telling you that the monomeric unit, they are different in heteropolysaccharide. Then we look at homo polysaccharide starch, which is number 1. Example: starch, there are 2 forms of starch: the amylose and amylopectin. The major glycosidic bond in it is alpha 1,4 glycosidic bond. Alright. So for amylose, you see the amylose is unbranched chain, long chain that is not branched, linked together by alpha 1,4 glycosidic bond. Okay, yes, they are water soluble and stained blue by iodine. But we have another aspect of starch, which is amylopectin. So starch has 2 types of polysaccharides in it: amylopectin and amylose. Amylopectin is the branched aspect. That one, amylopectin is the branch. Look at it here. Amylopectin, branched, contain alpha 1,4 and alpha 1,6, up to million glucose units. Water insoluble. Amylopectin is 80 to 82% water insoluble. Stained red brown by iodine. So you can see it. They have branch. Those branch is alpha 1,6. So you can see the branch here. This is where they usually branch. See the branch going up. See? So the bond here is usually alpha 1,6 glycosidic bond. I get it. So the bond here is alpha 1 cis glycosidic bond. This bond, alpha 1 cis glycosidic bond. Okay. So another type of homopolysaccharide is glycogen. The difference between glycogen and starch is that whereas starch is of plant origin, glycogen is found in animal. Is the storage form of glucose in animals. All right. So the both of them contain the same monomeric unit, glucose unit, and they are also branched. But another difference between glycogen and starch is that glycogen has more branches. It has more branches than starch. Okay, so that they can be easily mobilized. So glycogen has more branches than starch. That's just the difference between the two of them. So take note of that. Then we have we now also have what is called chitin. Another homopolysaccharide is chitin. The constant of chitin, the monomeric unit of chitin is usually N-acetylglucosamine. You see it? N-acetyl what? Glucosamine. The monomeric unit of chitin is N-acetylglucosamine. Chitin is the structural component of insects, the skin of insects. Okay? Yeah, so it's made of chitin. Look at it. See the difference between chitin and starch is that both of them have glucose, but the glucose in chitin is usually glucosamine. This is N-acetylglucosamine. It's different from chitosan. Chitosan is just glucosamine. Why is chitosan only glucosamine? Because the acetyl group is no longer present in chitosan. Look at chitosan. In chitosan, you have only the amino group present in chitosan. The amino group, look at it. But in chitin, you see it is N-acetyl group. So chitin is a polymer of N-acetylglucosamine, whereas chitosan is a polymer of glucosamine. Okay? Pectin is a polymer of galacturonic acid, as you can see. Alright? Then that takes us to heteropolysaccharide. Heteropolysaccharide composed of mixture of monosaccharides. On hydrolysis, they will yield a mixture of monosaccharide. Heteropolysaccharides are further classified into two types: neutral sugars and mucopolysaccharide. The neutral sugars there include hemicellulose, gum, mucilages, and pectic substances. They give more than one type of sugar unit on hydrolysis and sometimes non-sugar components. Alright? So, what of mucopolysaccharides? Mucopolysaccharides are heteroglycans made of polysaccharides made up of repeating units of sugar derivatives, namely amino sugars and uronic acid. Example is glycosaminoglycan, GAG. Then you have also these mucopolysaccharides. They are usually components of tissue structure. Some mucopolysaccharides are found in combination with proteins to form mucoproteins or mucoids or proteoglycans. Mucopolysaccharides include hyaluronic acid, heparin, diamantane sulfate, and keratan sulfate. So, example is hyaluronic acid or hyaluronate. This is a glycosaminoglycan with repeating unit of disaccharide consisting of two glucose derivatives, glucuronates or glucuronic acid and the N-acetylglucosamine. So you see, glucuronic acid and N-acetylglucosamine join together in a repeating unit. So two types of monosaccharides have joined together: glucuronic acid, N-acetylglucosamine. Okay, join together to form this hyaluronate. You see, the enzyme is called hyaluronidase. these hyaluronidase have the capacity to digest hyaluronic acid. Okay, that is one thing about snake venom. They contain hyaluronidase that have the capacity to digest structural component that's something that is made up of a hyaluronate. I get what I'm saying. So this is the structure of hyaluronate. Look at the two monosaccharide, D-glucuronate and the N-acetylglucosamine. Look at the glycosidic bond is between carbon 3 of N-acetylglucosamine and carbon 1 of glucuronic acid. This is carbon 3 of N-acetylglucosamine and then carbon 1 of D-glucuronate. See it? Carbon 1 of D-glucuronate and carbon 3 of N-acetylglucosamine. Alright. So, another type of mucopolysaccharides is keratan sulfate. Keratan sulfate is a heterogeneous GAG with a variable sulfate content besides small amount of mannose, fructose, sialic acid, etc., which essentially consists of alternating unit of D-galactosamine and N-acetylglucosamine. They are present in cartilages, bones, cornea, nails, hoofs, and claws. Look at it here. Galactosamine and N-acetylglucosamine. Look at it here. The linkage is beta-1,4 and also beta-1,3. So the two glycosidic bond here is beta-1,4 glycosidic bond and beta-1,3 glycosidic bond. You can see the sulfate. See the sulfate group. See the sulfate group? That is why it is called keratan sulfate. With, you can see that it has a galactosamine, N-acetylglucosamine. Then we have another type of mucopolysaccharide called heparin. Heparin is an anticoagulant. Look at it here. Heparin, an anticoagulant that occurs in the blood, lung, liver, kidney, spleen, etc., made in mass and released into the blood. Heparin is composed of alternating units of N-sulfo-D-glucosamine, C-sulfate, and glucuronate 2-sulfate. Look at it here. See it here. See? Very powerful compound. N-sulfo, glucosamine, C-sulfate. Look at the C-sulfate. See the N-sulfo. N-sulfo, D-glucosamine, C-sulfate. See the 6-sulfate. See the 6-sulfate here. You see it? This is the 6-sulfate. Then gluconate 2-sulfate. Gluconate 2-sulfate. Wow. Look at the gluconate 2-sulfate. Look at the 2-sulfate there. You see? Look at the 2-sulfate. Look at the 2-sulfate here. See the 2-sulfate. See the glycosidic bond is alpha 1,4. Glycosidic bond. Okay? So, it's very, very important. It's a powerful polysaccharide, mucopolysaccharide. Like we say, it's an anticoagulant. Thank you.